Good morning to one and all. Today's topic is on affinity chromatography. What is affinity chromatography? It is a biophysical technique, a type of liquid chromatography that enables separation, identification, purification of components from a mixture of substances. It involves both qualitative and quantitative analysis. It utilizes reversible biological interaction that refers to attracting force exerted by the different atoms that cause them to bind in combination. What is the principle of affinity chromatography? The stationary phase is the support medium onto which the substrate binds covalently so that the reactive groups are exposed for binding. When a crude mixture of substances is passed onto the column, the substance with the binding site binds to the stationary phase while all other substances are eluted in the void volume of the column. Once the other substances are eluted, the bound target molecules can be eluted by changing the pH, ionic strength, or the polarity conditions. Here, this picture depicts the elution of the molecules based on affinity. The first, the first column shows the matrix that are equilibrated with the wash buffer. The second column shows the different protein molecules that are to be eluted based on affinity. The third column shows the wash buffer which is used for elution for the unbound protein molecules. The bound protein molecules are eluted using the elution buffer by changing the pH or the ionic strength or the concentration of the buffer. Moving on to the steps that is involved in affinity chromatography, the first step is the column is equilibrated with the binding buffer. After equilibration, the sample is applied onto the column for separation. The elution is performed specifically by changing the pH, ionic strength, or the polarity or the concentration. Finally, the affinity column is re-equilibrated with the binding buffer. Moving on to the applications of affinity chromatography, separation of mixture of components. It also removes impurities and also purifies molecules. It involves in detecting in vitro antigen antibody reactions. It also detects single nucleotide polymorphisms. It also detects the mutation that are seen in DNA and RNA. Moving on to the advantages of affinity chromatography, it is highly specific and the purity is high. The matrix can be reused and it is easy to recycle. The yield is high. It also removes contamination such as proteases. Moving on to the last part, limitations of affinity chromatography. It is a time consuming process. It involves expensive solvents and it also requires physical labor, and the proteins usually get denatured if the pH is not adjusted properly. These are the references that are used for the study, and thank you.